In our video for part 1, we described how the adaptive and innate systems are very much interlinked, very different to the old idea where they were acting independently of each other. In this video, we're going to look at the adaptive system in more detail. But one of the bridges between the two systems are the phagocytic cells that we've talked about several times in the course. And basically, if we look at the way that a typical phagocytic cell works, you can see that the large blue cell just represents an immune cell. It could be a neutrophil, although the nucleus is the wrong size and shape for that. But the basic idea is that in step 1, what's happening is that the bacterium is attracting the neutrophil or the immune cell, or maybe the other way round, the immune cell is detecting the bacterial cell. The immune cell closes in on the bacterial cell and starts to surround it in step 2 with extensions of its cell membrane. And that process continues in step 3 until the bacterial cell's completely surrounded. And it's now enclosed in a little vesicle made of the cell membrane of the immune cell. Once that happens, lysosomes become attached to the vesicle and inject toxic and destructive chemicals in, which completely destroy the bacterial cell, which is then voided out into the surrounding tissue fluid by the process of exocytosis in step 6. So what we're going to do is we're going to look at the number of ways that these phagocytic cells actually become involved with the specific or adaptive response. We're going to look at the way that two groups of lymphocytes, white blood cells called lymphocytes, work. We're going to look at B lymphocytes, which are released from the bone marrow, and they take part in something called the antibody response. They can also multiply by themselves because they have a nucleus, unlike red blood cells. And the T lymphocytes are also immune cells in the bloodstream, and these are stored and released from the thymus gland, and they take part in something called the cytotoxic T cell response. So let's have a look at how B lymphocytes work. Let's imagine that a bacterial cell enters the body, and the B lymphocyte will basically recognize the antigen, which is the distinctive protein signature on the outside of the bacterial cell. And the B lymphocyte will recognize that as non-self. And it will actually recognize it in detail as to what type of bacterial cell it is. Once that happens, the B lymphocyte is triggered into very rapid multiplication. So very rapid mitosis takes place. And this gives rise to a line of specialized B lymphocytes that we call plasma cells. So these are differentiated and they will contain the machinery that's needed to go to the next stage. And the next stage is that the plasma cells produce vast amounts of protein, and we call those proteins antibodies. So antibodies aren't living cells, they're protein. And those particular proteins have a similar or matching shape to the proteins on the outside of the bacterial cell. what is also produced is something called a memory cell, and that will store up the memory of this encounter so that the recipe for that particular antibody protein is stored in the memory of the immune system and can be produced extremely rapidly. So the first time that the immune system encounters a bacterial cell, it may take anything between 5 and 7 days for the immune system to actually learn what shape of antibody protein is needed. The antibodies themselves actually have various mechanisms by which they result in the destruction of bacterial cells or the incapacitation of viruses. And you can see in panel 1 here, the Y shape just represents the shape of the antibody. And you can see that the antibody proteins have become attached to the swimming tails of the bacterium in picture 1. That immobilizes the bacterial cell, and the stem of the Y actually acts as a signal which attracts phagocytic cells such as neutrophils to the site of the encounter, and the bacterial cell along with the antibodies are then engulfed by phagocytosis and destroyed. You can also see that the antigen is actually binding to the outside of the bacterial cell. So if you have bacterial cells without swimming tails, the same thing happens. Panel 2 shows a slightly different mechanism, and this is very much simplified, but essentially what's happened is that the antibody protein attaches to the bacterial cell membrane. That induces the cell membrane to become permeable. Water rushes in by osmosis into the bacterial cell and bursts it. And also that stem of the Y on the antibody will attract in the neutrophils, which will come in and do the sweeping up and get rid of the residues. In step 3, we're looking at a virus which has become surrounded by antibody molecules, and therefore it cannot enter. And if you come along to the COVID-19 presentation that I'm doing in a few days' time, you'll hear a bit more about how viruses can actually be dealt with. Step 4 involves a process called opsonization. So when that stem of the Y acts as an attractant to other immune cells, it's a sort of marking process. We call it opsonization. And in this particular case, you can see that the little bacterial cell has got a couple of antibody molecules on it, which have attracted a pretty hefty looking neutrophil. Notice the funny shaped nucleus, by the way. And that's basically enclosing the bacterial cell and will lead to its destruction. We're not going to talk about antibody presenting cells or antigen presenting cells in this presentation, by the way. Okay, so let's just stand back and have a look at the different players in the immune system. So if we imagine this is a typical body cell, there's a nucleus inside it containing the DNA. By comparison, the average bacterial cell would be about that size. And then over there on the right-hand side, you can just about see a little red dot. That's a virus. Remember that viruses are not classified as living, despite the awful havoc that they can wreak. When you take a virus into your body, it can't move on its own, it can't reproduce on its own, it can't produce energy. How do they actually cause so much damage? Well, let's imagine that this red blob is a virus. The virus, let's say, enters the airways, and as you breathe in and out, the virus is being moved up and down, and eventually will actually collide with one of the membranes of one of the cells. And this is purely a chance process. And what then happens is that you get a sort of fusion event, and the virus fuses with the cell membrane. That induces the membrane to actually allow or take in the virus into the cell. So the cell is now infected. Once inside, the virus undergoes a process called uncoating. And essentially a virus consists of a protein shell, and within that shell you've got the genetic material of the virus. It could be DNA, it could be RNA. Now there may be other things in the viral outer coat as well. You may have some lipids, but for now just think of the virus as uncoating. So the genetic material of the virus is now loose in the cell. The host cell mechanism then, it's actually the cytoskeleton that does this, draws the genetic material of the virus into the nucleus, and once in the nucleus, the viral DNA becomes spliced into the DNA of the person that's become infected. And that seems to inactivate the DNA of the infected person to a large extent, and switches the cell mechanisms over to making proteins, viral proteins. So what actually happens is that the cell very obligingly starts manufacturing lots and lots and lots of proteins called viruses or virions, and eventually it gets to the stage at which the actual cell bursts, and it will release the virus out into the bloodstream or into the airways or wherever it happens to be. And it could be done by two processes. Different viruses work in different ways. Some of them basically leave the cell alive, the infected cell alive, but obviously infected, and release the virus particles in little vesicles, a process called budding. Other ones completely wreck the infected cell, and that's a process called bursting. Now, of course, the big problem with this is that the virus has tucked itself away, and that's a very bad way to describe it. It's become enclosed in a body cell. So the B lymphocytes, unless they're extremely quick, don't cotton on until it's too late. This is where the T lymphocytes come into play. And we're going to just compare the way that B lymphocytes and T lymphocytes work. So there's our B lymphocyte that we talked about a few minutes ago. And over on the right-hand side, we're starting to build up a flow diagram about T lymphocytes. And again, there's a process of recognition here. So the T lymphocyte recognizes and binds to the viral antigen. In other words, the distinctive shape of the proteins on the membrane of an infected cell. So one of the things that happens is that when a virus infects a body cell, it will usually leave some of its viral protein incorporated in the cell membrane. So it's a bit like a burglar breaking in through a window, cutting his hand on the glass, and leaving a little specimen of his blood there. And Sherlock Holmes will actually be able to identify who the burglar was. And in this case, the T lymphocytes recognize that viral antigen. And rather like the B lymphocytes, they multiply like crazy. But not to form plasma cells, they form distinctive T lymphocytes called T killer cells. T killer cells are quite nasty, and they will destroy virtually anything unless they're trained properly. And that training takes place in the thymus gland, where they're bench tested before they're released out into the bloodstream. The T killer cells have complementary receptors, and they will destroy any infected body cells. So any body cells infected with virus will actually be destroyed and picked out. The T killer cells also produce memory cells, and they also produce cells called T helper cells. And the T helper cells work with the memory cells, and should that same virus enter the body again, the T helper cells mount a huge response. They act as a sort of central office, if you like, for the immune system. And the T helper cells will actually also prime up the B lymphocytes. So the B lymphocytes, this time, they get some warning that they need to get cracking and produce antibody for this particular virus, at the same time as the T helper cells are priming up the T lymphocytes. So they're the two wings of the white blood cell response, the lymphocyte response. And you can see how they work with the phagocytic cells, and also work with other substances called complement, which we won't be talking about in this video. We'll have to go a bit deeper into that in a separate unit.